Welcome to Study Loop.
Okay, in order to answer this question, we have to talk about the GRAS protein, okay? Maybe this is your cell. Here you're going to have your GRAS protein. Okay, let's stay it like this. Your GRAS protein. Here you're going to have your tyrosine receptor, okay? Receptor for tyrosine kinase. And here you're going to have your ligand. When your ligand is not present, then this receptor normally is not going to be active, and GRAS is going to bind GDP. And this means that the gas is in an inactive state. Okay, but what happens when a ligand binds here? For example, insulin it acts on a receptor for tyrosine kinase. Well, once a ligand binds, GDP is going to be removed, and GTP is going to bind gas, and this is going to start a cascade called MAP kinase cascade. Okay, that it is going to lead to changes in the nucleus in order to stimulate cell division, cell growth. Okay, but how or well, this gas protein, as it causes this, if there is too much activity of this gas protein, it can lead to too much cell division and also cell overgrowth, and this is very dangerous because it can lead to cancer. Okay, so this is why the gas protein, besides the action of tumor suppressor proteins that are going to regulate the gas in order for it not to become too active, the gas protein itself has a GTPase, an intrinsic GTPase domain. Okay, then when the cell does not require more of the gas action, this GTPase is going to act on that GTP and it is going to make it bind GDP again, so the gas gets inactive. Okay, this is how the GTPase, the intrinsic GTPase, works. So the question says, the gas protein is a G protein connected with a response to receptor tyrosine kinases that initiates the MAP kinase cascade when GDP is released and GTP uploaded. Mutations in the gas protein which interfere with its GTPase activity, these are common in cancer. Evaluate the connection between the inability of gas to hydrolyze GTP and uncontrolled cell proliferation. Okay, option A says gas, when bound to GTP, becomes permanently inactive. This is totally false. Okay, it would become totally active, permanently active. Option B says gas, when bound to GTP, okay, when GTP becomes permanently active, this is correct, even in the absence of the ligand, it no longer regulates cell division. This is so true. Okay, remember that gas becomes active and binds GTP when the ligand binds to the receptor of tyrosine kinase. Once the ligand is not there, then the GTPase is going to break down GTP into GTP, and the gas protein is going to become inactive. However, when there is a mutation of this GTPase, and this GTPase is not going to act even when the ligand is not there anymore, and the gas protein is going to continue like bound with the GTP, and this is going to cause a permanent activation of this cascade and a lot of cell division and cell growth, and this is going to lead to cancer. So option B is the correct answer for this question.